introduce our speaker, who is Georgia Dubeux. Georgia is a master's student here at UF in the Animal Sciences group. She is from Brazil and holds degrees in medicine from Brazil. She has a lot of experience in a variety of different things. She has experience in pediatric medicine. She's worked with bacteria and oysters. She has a lot of experience in genetic analyses. And again, her degree that she's working on right now is in animal sciences. And she's going to be talking to us about Gracilaria, which is a macroalgae that's very abundant in our region, and how you can feed it to cows as a feed additive. This is something I know nothing about, and I'm very excited to hear about it. So I'm going to stop sharing, and I'm going to turn it over to Georgia. Thank you for being with us, and I'm excited for this presentation.
Thank you, Dr. Reynolds, for the introduction. Let me share my screen here. Okay. Can you guys see me? Can you guys see me? Yeah, good.
That's great.
Thank you, Dr. Reynolds, for the introduction. I'm very excited to be here. And yes, we are working with this algae species Gracilaria, and we are very excited about it. So let's start. I'm going to start talking about the global temperature. We know, and some of us can feel actually, that the global temperature is increasing, right? Since the pre-industrial times, when the fossil fuels started being burned for energy, we are having an average of 0.06 Celsius degree per decade in the increase of temperature level. 2023, 2023 was the warmest calendar year in global temperatures data record since 1850. And it was 1.5 Celsius degree above the pre-industrial level. So now we have the last 10 consecutive years, the 10 warmest years ever recorded. And I think everybody knows what's going on, right? We have the greenhouse gases. So the greenhouse effect is very briefly and basically, the sun's energy reaches the earth, and part of this energy bounces back to the space, and part of the energy, the rest of this energy, is absorbed at the surface, Earth's surface. So then the Earth's surface will send back energy in form of heat back to the space. Some of this energy leaves the Earth, and some of this energy is trapped here in this layer that the greenhouse gases make and trap this heat. The problem now is the human activities has been releasing so much greenhouse gases in such a speed that these greenhouses here are not allowing the heat leave the space in the amount that it should have, you know. So we are having an increase in temperatures. We have a measurement for greenhouse gases. It's a global warming potential, GWP. So we have for carbon dioxide, the GWP 1, for methane 28, and nitrogen oxide 265. And this measurement, it basically, measures the amount of heat trapped by a certain mass of gas and compare this with the carbon dioxide. Meaning, for example, that 1 ton of methane is equal to 28 tons in CO2 equivalent. And this is important because we can quantify the relative impact of different gases on climate change. But I have a question for you guys. Are ruminants and their waste the major contributors to greenhouse gas emissions? Are they guilty, right? So here in US, we have 74% of greenhouse gas emissions in carbon dioxide. 11% is methane, 6% in nitrous oxide, and 3% in fluorinated gases. All economic sectors, they contribute for these emissions. So we have transportation, 29%, electrical power industry, 25%, industry, 24%, and you have agriculture, 10%. I put this figure on purpose here because they represent the agriculture sector as a cow and put 10%. It's kind of misleading. But actually, the livestock sector is responsible for 3 to 4% of the total greenhouse gas emissions. And from these 3-4%, 90%, around 90% is methane from ruminants. But what methane in ruminants has to do, right? Well, the ruminants, they have a very, very special stomach. It's divided in 4 compartments, and the rumen is the biggest one. This rumen has a very unique microflora that evolved with these animals for millions of years, and they have a symbiotic relationship. So the animal eats the plant material, but he cannot digest the fiber, but the rumen microbes can. So they will digest the fiber and will provide the animals with the volatile fatty acids that the animals will use as a source of energy, and also the microbials you use as a source of energy. As a result of this fermentation, we're going to have CO2 and hydrogen. So, but hydrogen is not good for the rumen environment. So we have special bacteria, the methanogens, that we will get this hydrogen combined with carbon and make methane. And the methane is going out from the animal. But it's going out from the mouth, 90%, and not from the ear, just ear part of the animal. Just 5% of methane goes from the rear part of the animal. But this methane is very, very important also to us as nutritionists, because it represents a loss of energy from the ruminant, from the animal. So 2 to 12% of gross energy, it's lost in methane production by the animal, right? So our goal is save this energy, reduce this methane, and make this animal more energy efficient, and more productive. There are many strategies to mitigate methane in an animal. Since breeding, because we know there are some animals that they emit less methane. So we try to select these animals and breed them and spread them. It's a way to mitigate methane. Also management in the pasture, putting like legume in the pasture. Everything makes this mitigation of methane and have the objective to improve animal production, improve feed efficiency. So we also have feed additives, right? Feed additives are substances that we add in the diet of the animal with the intent to get a better digestion, a better fermentation, and target all the fermentation processes in the rumen, all the microbial composition in the rumen. So I put here 2 examples of chemicals that is used. One is the 3-NOP. 3-NOP is very effective in inhibiting methane, and it blocks one of the steps in the chain in methane production inside the rumen, and it was approved by FDA last year. Also, we have the bromoform containing seaweeds, So these seaweeds have this chemical that has a great ability to reduce methane. And some studies shows that it can reduce even 80% of the methane production. So why we are studying algae, right? So we know that the red algae might have bromoform. Also, they can have saponin and tannins. And all these compounds have the potential to reduce methane emissions. Also, Florida's climate and coastline length provide a unique opportunity to explore the potential of algae as a cattle feed additive. So how everything started? Well, this is Aaron. Aaron is an oyster farmer in Tampa. He owns the 2 Docks shellfish. And he was noticing that a red seaweed was growing around the ropes in the cage that he uses to cultivate his oysters. So Aaron also has a PhD in environmental sciences. So he identified the species as Gracilaria and did a little research and saw that all the species of algae has been used around the world for many things. So he contacted us and said, hey, what we can do about it? So we came up with this question. How can we develop a solution to assist the oyster farmers while utilizing algae as a beneficial feed additive for cattle? We have a lot of research with another type of red seaweed, is another species, Asparagopsis. This Asparagopsis has a lot of bromoform, up to 5% of its biomass is bromoform. And they are feeding animals, and also they have in vitro studies showing the reduction in the methane production in the animal. So our rationale is that there are previous research showing that bromoform-containing algae can alter rumen fermentation and reduce methane production, but Florida algae have not been fully explored. But what's bromoform, right? Bromoform is a chemical, is a halogenated compound. It's colorless at liquid temperature. It's very volatile and unstable, and it's toxic. Actually, bromoform is considered carcinogenic because it can cause cancer in rats. So it's considered carcinogenic for humans too. And how the algae produces bromoform? Well, the algae has what we call reactive oxygen species that is produced inside the algae cells in metabolic processes while they are doing photosynthesis, respiration, or responding to some environmental stress. So I represent this reactive oxygen species as oxygen peroxidase is one of them. But also this oxygen, I will call ROS, is important to the algae because they work as signaling to control also other processes in the algae, like growing or development, even to put around the cells to respond better to changes in the environment. So too much ROS can also damage the algae cell. It can damage the membranes, it can damage the protein. So the algae, in order to balance these products inside them, they uptake bromine from the seawater. This bromine gets inside the cells, and the algae has a very specific enzyme, it's vanadium bromoperoxidase, that is going to catalyze this reaction, and it will form an acid. the hipobrom acid. It's here. So after this acid is formed, it's going to pass into a methylation, and bromoform is going to be result from this reaction. So once this bromoform is formed, the algae will accumulate inside their cells, and we use the main role is for defense. So the bromoform play a very important role against predators like manatees and other competitors. Algae that has bromoform are considered more aggressive. But besides its toxicity for humans and rats, it seems to be harmful for the animal, the cow, right? There are a lot of studies showing that the animal has no toxicity, does not show toxicity with this component. And also, when the amount is not too high, the level of inclusion is not too high, no bromoform will accumulate in the tissue of this animal, and also will not be released by the milk. Okay, so it's safe also for the humans. And the bromoform is metabolized inside the rumen, inside the bacteria that makes the methane. So the animal will eat the algae. This bromoform is released inside the rumen. So it's going to be incorporated inside the methanogens, the bacteria that makes the methane. And then inside this bacteria, it will compete with the active site of two enzymes very important for methane formation, the coenzyme aminotransferase and the methyl coenzyme aminoreductase. And it's going to inactivate these enzymes. So in the chain for methane formation is going to be blocked, and we won't have methane. In some studies, it's up to 80% this reduction. So our experiment 1, we have the objective to explore how Gracilaria species and drying methods could affect methane mitigation on ruminal in vitro fermentation. We decided to do that because, as I told you before, the bromoform is volatile, but you want to see if you have a difference with the sun-drying the algae. That is the most common thing around the industries around the world for treating the algae. So we decide to freeze dry first the one part of this algae. That means that we're gonna get the frozen algae, put inside a machine, and then the ice is going direct to vapor without passing for the liquid phase. So there's no volatilization. And the other algae was sun-dried. So we went to Tampa to harvest the algae. The first thing that we did, we put the algae inside Ziploc bags. We put in a cooler with ice. And then when we arrived at the shore, we put in dry ice. It transported to Mariana, to NFRC. Here in our lab, we decided to wash this algae because the algae comes with a lot of sand and small creatures. So we decided to wash the algae. So the next day I stopped freezing dry, and then I wait for the sun-dried algae to arrive. So for this first experiment, we had 2 Gracilaria species, the Gracilaria mamillaris and the Gracilaria damicornis. Those species were identified by Aaron. So the algae were freeze-dried or sun-dried, and we tested in 4 inclusion levels: 0, that was our control, no algae added, 1, 5, and 10% on dry matter basis. And we used the Bahia grass as a basal diet. So we performed 3 batch cultures, right? And we measured the responsible variables: in vitro organic matter digestibility, VFA, that's volatile fatty acids, ammonia nitrogen, pH, total gas, and methane. So my treatments were the 2 species of Gracilaria. They were sun-dried or freeze-dried. And in my tubes, of course, all these tubes were in replicates, and I had 0, 1, 5, and 10% of inclusion level of my algae. For those that are not familiar with what a batch culture is, basically we try to simulate what happens in the rumen of the animal, but in the lab. So what we do, we first adapt this animal for 2 weeks in the basal diet. In my case, was the Bahia grass. And after these 2 weeks, we collect rumen fluid from these animals. We use 2 animals. So we mixture this rumen fluid in equal parts. We mix with a buffer, you know, and everything until this stage is very fast. With control of temperature, we keep the temperature of the system 39 Celsius degree and a constant flux of CO2, because the rumen is anaerobic and the exposure to oxygen would kill the microbes. So we incubate for 48 hours. And after that, we collect material for ammonia. We measure pH, we collect material for VFA, and we measure total gas and methane. Another part of our tubes, we do the procedure like the real stomach of the animal. So we add pepsin and hydrochloric acid and incubate for more 48 hours. And then we're going to determine the digestibility. So we start with our results. We saw that the Gracilaria has a low concentration in crude protein. It was expected because we have this in the literature. The organic matter is very variable. We watched the algae, but even though we had a big range in organic matter, but our algae did not have bromoform. We were expecting that in the algae that was sun-dried because of the volatile compound of the bromoform. But we were expecting the Gracilaria that was freeze-dried had some, but the amount was too little and the lab was not able to quantify, was below the limit of quantification. We were was disappointed a little bit with that. So we also studied the heavy metals, and we saw that for arsenic and lead, the ones that are everybody gets concerned because algae can accumulate these heavy metals. They were good below the maximum level permitted per kilogram of diet of the animal, but the cadmium surpassed the level, was higher, in the 10% level of inclusion. So, but we think that in lower inclusion level would not be a problem. So going to methane, we saw that two Gracilaria species had results, linear decrease for methane for the dry matter incubated, a linear decrease, significant, was Gracilaria mammillaris freeze-dried and the Gracilaria damicornis sun-dried. In the treatment, Gracilaria mammillaris sun-dried and Gracilaria damicornis freeze-dried, we did not have results. We were happy with that because we did not have the bromoform, but even though we had reduction in methane. So when we correct this dry matter incubated for organic matter fermented, I mean, the actual particle that was digested by the microbe, we also had a tendency decrease for the same algae, which was really good. So for the other results, what we had was for the in vitro organic matter digestibility, we did not have any response, any significant results for any treatment, which is really good because usually the feed additives that reduce methane, they decrease the digestibility in the animal. So the fact that we have something that reduces methane and doesn't change the digestibility, it's good. So we had a linear decrease of pH for Gracilaria mamillaris sun-dried, Damicornis sun-dried e a Damicornis freeze-dried, e a quadratic effect for Gracilaria mamillaris freeze-dried. But the range pH was very, very small, and we think there's no biological effect in the animal. For total gas, we had a reduction for treatments of Gracilaria mamillaris freeze-dried, Damicornis sun-dried, and Damicornis freeze-dried. No effect in ammonia nitrogen. We were expecting that because the algae was not rich in protein, right? So there's no meaning to change the ammonia nitrogen. The total VFA had a linear decrease for one treatment, the Gracilaria damicornis sun-dried, and had some changes in the volatile fatty acid VFA profile, but we did not see nothing that could be biologically important for the animal. So the conclusions for our first experiment was that Gracilaria mamillaris freeze-dried and Damicornis sun-dried have the potential to reduce in vitro methane production without reducing in vitro organic matter digestibility, even without bromoform. And heavy metals might be a concern depending on the inclusion level. But we want the bromoform. We want to test our algae and see if it has bromoform. So we analyzed our protocol, and we saw that maybe the fact that we transport to an FRC the algae and wash the algae here, put in ice and not in dry ice, everything together probably made us to lose our bromoform. So we improved our post-harvesting protocol. So we had to harvest, we collect the algae, and immediately after collecting the algae, we wash the seawater, and we snap freeze with liquid nitrogen. So then we put in dry ice at the shore, and then we transport it to the NFRC. We did not wash the algae. I put in the freezer -20, and the next day I start freezing drying. So the objective of the second experiment was to explore the potential of the same Gracilaria, but the potential, we wanted the algae with bromoform in methane mitigation. And this time we had bromoform, right? We were very excited about it. The amount was very small when compared with the other species, the Asparagops, but yes, we had bromoform, and we are very excited. So the second experiment, we add one more Gracilaria species, a Coronopifolia. All algae were freeze-dried. We run 3 batch cultures the same way, and we measure the same response variables. The batch culture, we did exactly the same as the first one, and I explained to you guys. And we analyzed the new species for heavy metals, and we had the same results in cadmium, right? The protein was a little bit better. Arsenic and lead was not a concern, but cadmium was. So for methane, we had a reduction in methane per gram of dry matter incubated for 2 algae, Gracilaria damicornis and Gracilaria coronopifolia. We had a linear decrease with the increase of inclusion level. But we did not have significant results for Gracilaria mamillaris. What got us surprised, in a surprise, because the Gracilaria mamillaris was the algae that had more bromoform. So when we translated these results to organic matter fermented, we also had the same responses. We had a linear decrease in Gracilaria damicornis, and Gracilaria coronopifolia was significant, but no significant effect for Gracilaria mammillaris. And then for the other variables, the results were pretty much the same as the first one. We did not have changes in pH this time and not changes in the total VFAs. But for our surprise, this is a correlation table. Bromoform had nothing to do, any correlation with any methane results or any other variable. And we're like, what's going on here? So we saw as a conclusion that Gracilaria species have the potential to reduce methane production in vitro studies, even without bromoform. The reduction had no correlation with bromoform. But of course, in vivo studies should be done to confirm these results. But if it was not bromoform, what else can be, right? So as I told you before, Gracilaria, the red seaweed in general, is rich in saponins, tannins, unsaturated fatty acids, other compounds that can also mitigate methane. But unfortunately, we don't have these studies with us. We did not study these compounds in our research. So other chemical compounds and their effect in methane production weren't further studies. And several aspects still need to be investigated before taking red seaweed Gracilaria species to the market, but I think it's a beginning. So with that, I want to say thank you to my advisor, Dr. Nicholas Di Lorenzo, and my committee members, Dr. Diwakar Vias and Dr. Ashley Smith. I want to say thank you to my amazing nutrition team in Dr. Di Lorenzo lab, the Florida Carol Enhancement Board, and CITIT. So with that, thank you, everybody. And let's go to the questions.
Thank you so much. That was really interesting and all new for me, and I really enjoyed it. We already have a question, and I'll read that. But folks, if you have questions, please put them in the chat, or if you'd like to unmute and ask your question directly, you can as well. The first question is, do you think that the cadmium content of the Gracilaria could be more due to the system in which it was grown? If it was grown in a different water body, would it have lower cadmium?
I'm sorry, can you repeat this question?
Yeah, so cadmium was an issue. It's in the chat if it's easier to read.
Okay, you're asking if it can be a concern with the cadmium?
Or if you grew it somewhere else, maybe there would be less cadmium in the.
Okay, yes. All heavy metals are very variable in all algae, you know. They have all algae in general, the red seaweed and the green seaweed also have the ability to accumulate these heavy metals. And we don't know why all our algae was rich in cadmium. Maybe the Tampa water was a problem, right? But when I explained there in kilograms of milligrams per kilograms in dry matter, it's in kilograms of diet. So if the animal eats 10 kilograms of diet, the cadmium will be 5 milligrams, you know? So if I use in the whole diet, right? So if I use like 5% of inclusion level, I won't reach this level. So it won't be a problem, you know? But yes, the cadmium, arsenic, and other heavy metals are very variable. And it's a concern because we have to test these heavy metals because they can accumulate in the animal's tissue and can pass to the human food chain. Yes.
There's another question in the chat. Do you have any plans to test Gracilaria vermiculophylla in the future? That's the invasive one, right?
No, for now we don't have plans to going on with the Gracilaria. Yes, we're going to keep our Gracilarias, the ones that we have already.
Yeah.
Yeah.
I had a question sort of related to that cadmium question. How do you expect like the bromoform or any of these secondary compounds that might be causing the reduction in methane. Do you expect that to vary by location too? Like if I grew the Gracilaria in a more stressful environment, would you expect those compounds to differ in their concentration as well?
I think so, yes. But what I've been reading is the algae has a very efficient system to keep these the ROS, we call the reactive oxygen species, under control, you know. But of course, if they have a lot of environmental stress like UV light or pollution, they will have more bromoform accumulating their cells. Yes, that can happen. Yeah.
Cool. So you need to stress these guys out.
Yeah.
I don't see another question pop up, so I have one more, a sort of follow up. And maybe this is a silly question, but how much does a cow eat in a day? Like, how much would 5%, like, if you wanted to feed it, and no, we're not there yet, but if you wanted 5% of the diet to be Gracilaria, how much would that be?
Well, for example, the cow eats, I believe, around 2.5% of the body weight per day, right?
Okay.
So if I have a 500 kilograms, I will have a cow that will be 10 kilograms, right? So if I want to put a 10% level of inclusion, I will put 1 kilogram in the diet and 9 kilograms of the 9 kilograms of diet, 1 kilogram of algae. Is that how it works?
So you need a lot of algae.
Yes, it's a lot of algae. It would be a very profitable market if it's working, I think.
Yeah, yeah. Any other questions from the audience? There's another one in the chat. How much would something like this increase the cost of food? Is it cheaper than the other compound that is already approved?
Well, that's a good question also. We don't know because we don't have this market yet. But for sure, we need to do a study how much would be to scale up this production, right? I think one big thing right now is the freezing drying method. That's an efficient method to keep bromoform, and it's going to increase the cost. But has another method around that is new. They are preserving the algae in canola oil, in vegetable oil. It seems to be efficient, you know. So all these alternatives must be studied and be considered if you want to a profitable product. Yeah.
I have one more question. You picked Gracilaria, and it sounds like you sort of picked that out of the idea that the oyster farmer found a lot of Gracilaria, but you know, would other red algae be, is there a reason that you picked Gracilaria beyond that, or would other red algae be just as, potentially just as, or even more efficient?
Well, we don't know yet. We
Yeah.
start working with because of Aaron. Everything started with Aaron. So he had the algae, he identified the algae, and we got interested about it, you know. But there are a lot of species of algae in Florida. Gracilaria seems to be the one that grows more in this area here. And there are some few farmers already cultivating algae here in Florida. But yeah, maybe there are other algae with potential also.
Great.
Yeah.
Thank you. Anybody else have any more questions? All right. Well, I think we'll end there. Thank you so much. This was a super interesting talk, and I learned a lot.
Thank
So I
you.
appreciate you being here.
Thank you very much. I really, really enjoyed to be here.
Thanks. Hope to
Bye.
see you all in our next Nature Coast Currents.
Bye bye. Thank you.
Thank you.